In every Banff meeting, one of the most important days is the first day where we have a day-long symposium on something. And in 2019, it's going to be on regenerative medicine, tissue engineering, pathology. So regenerative medicine, we're going to talk about that a lot. And it's an area that is the greatest promise for the future of medicine. It is also where the greatest intellectual dishonesty, fraud, stem cell tourism, and stem cell hype, all that. So you have to sort of keep all those conflicting ideas in your head. And this is, in fact, one of the origins of this initiative that we have called The Future and All That Jazz, where we insert important scientific ideas into music and poetry that people would never sit still to listen to a lecture on. If you advertise such a lecture, they suddenly discover they have to wash their hair or something important conflicting, and they can't go to the lecture. But if you entertain them with poetry and music, you can get them to think about the same things. We're providing organs only to 10% of the people who require them. And most people die waiting. I mean, that's the simple answer. I know that it's a wonderful advance. I'm proud to be part of it. But it could be tenfold or more better, where we are providing organs to everybody who needs an organ. That's what stem cell generated organs, stem cell repair, regenerative medicine, tissue engineering, that's the promise that it could provide. And it could do a lot more than that. It's not just transplantation, it's whatever is your area of greatest interest. Regenerative medicine could also provide remarkable benefits there. So in 2013, we created a nonprofit Swiss foundation, the Banff Foundation for Allograft Pathology. So I'm the chair of that. You can be the chair for 2 3-year terms. So my term is up in 2019, and Michael Mangle will take over from me then. We plan ahead very far. We were just talking today about the 2025 meeting. I mean, we know exactly where all the meetings before that are, but 2025 we just decided would be in Asia. It must be, because so many people from Asia have been coming to our meetings, it's only fair that 2025 be in Asia. Dr. Sun proposed an hour ago that it be in China. I think it's a very good idea. So I suspect that you're the first audience to hear that the 2025 Banff meeting will be in China. So you may know that this institution here at the University of Alberta is the 6th ranked institution in terms of transplantation research. So how do you think those rankings work? Do you think that they send people to every university, have them stay for 6 months and talk to people, interview people, and assess the quality of transplantation science in every university in the world? Nah, they don't do that. They just look at literature citations, right? So we are the 6th best in the world in terms of literature citations in transplantation. And the Banff process is the major reason for that. We just had the first full-length article on tissue engineering Banff classification accepted by AJT. And so there will be more publications now, because we're now talking about 2 different classifications. We have the existing one, we have the new one. They'll be concurrent for a long period of time. And is Canada ready for this? Does the Canadian public want this? Yes. There's evidence that the average Canadian voter and taxpayer likes the idea of radical life extension and regenerative medicine. And it is within medicine kind of the analog of the technological singularity, which I talked about earlier, is that point in time when machines are smarter than we are. Technological advance accelerates very quickly. We can only understand what's going on in the world if we collaborate or merge or cooperate with sentient machines in some way. So that may be hard to wrap your head around, but something closer to home is that regenerative medicine is kind of the medical part of this. So what do decellularized, recellularized organs look like? First of all, it's hard to get a situation where you do that and get an interesting result at the end. So Harold Ott, who runs the lab this work comes from, this is from a study by Song et al. In 2013, describes exploding kidneys, trying to infuse with the proper pressure to get enough cells in. And a lot of the kidneys simply explode, and a few don't, and you end up with a large number of cells in there, and then you can see what they do. But it's very hard to make it a reproducible procedure. It isn't. It still sort of catch can in terms of getting the cells in there. And so people who are skeptical about regenerative medicine say, well, can you call this a kidney? Well, of course you can call it kidney. It has tubules, glomeruli, what else? Yeah, so of course it is. And it's not that you should criticize it and say it's not a kidney. You should figure out how can we make it better? How can we make the process of putting the cells in better? Astic Petrosyan, who's kind of a young person, the world's expert in extracellular matrix in the kidney. She has been doing studies that's entirely human. You take stem cells from human amniotic fluid and you infuse them into basically discarded human kidneys that cannot be used for transplantation and have been decellularized. And then you let them incubate for a while, up to 6 weeks, 8 weeks, and you see what you have. So like we ask in the paper, we say, we're not sure that these have differentiated into podocytes yet. Well, obviously they haven't, but maybe they have some beginning characteristics. Some of them are in the right place for where podocytes should be, but others of them, just like in the rat, are wandering around within the interstitium. What are those cells actually doing? Are they dangerous? Are they headed in the right direction if you let them incubate for a few more months? Will they turn into something useful and know the right places to go, or will they create a teratoma? So there are lots of interesting questions. The question is, do we need long loops of Henle? Do these kidneys, these stem cell generated kidneys, need to be able to concentrate the urine? Because that is tough, because when you make stem cell generated organs, they tend to make shortcuts. And so the stem cell generated kidney would like to have short loops of Henle entirely and leave out the whole concentration pathway. So what's interesting, Harold Ott, if you listen to we had him speak in the course that I gave, and we took sort of the best 6 or 8 minutes of that. And what he talks about is the regenerative medicine organ as a device. Think of it as a physical device. And what do you want that device to do? What features should it have? Probably you can't pick everything that the normal kidney does or normal other organ does, because that's going to be decades before we can produce those kinds of perfect stem cell generated organs. So maybe a kidney that can filter waste just great but can't concentrate the urine, maybe that's just fine. So that's another way of thinking. He is Associate Professor of Surgery, Director of Transplant Biology Research Center, and my boss.
Today, I'm talking about the topic of transplantation tolerance. Probably you would think that transplantation tolerance is not its own topic, It's probably nothing new, and probably seems not exciting at all. But I hope in next 45 minutes, you will learn something new, and you will know something you have never heard before. And as a transplantation biology lab, most of our studies actually in animal models. The animal models we are currently studying including mouse, rat, pig, and monkeys. And I should acknowledge that these animals are truly heroes in our discovery. 90% all transplant recipients survive in the first year. However, the long-term outcomes have remained suboptimal. For example, only 44% kidney grafts are functioning at 10 years. In addition, the need for taking a lifelong immunosuppression has significant toxicities. Conversely, the tolerance, the success of transplant an organ without real lifelong immunosuppression has been the central goal of transplantation research in the last 6 decades. So, because the limited access for these protocols has raised the question, tolerance, is it worth it? The goal of tolerance for patients is to produce a reliable, non-toxic method for immunosuppression-free transplant success. So obviously, we are not there yet. So why aren't we there? Current knowledge suggests transplant may successful 3 different ways. First, imagine that there's no interaction between the recipient and donor graft due to effectively supplied immunosuppressive drugs. Second paradigm of recipient and donor graft interaction is that in which recipient takes on some of donor's properties, such as hematopoietic stem cells, to induce mixed chimerism, and that leads to some of patients withdraw from immunosuppression. But a third potential interactions between donor and recipient is that recipient repopulate of donor graft. So if that happens, then the donor will become recipient themselves, no need for immunosuppressions. So based on these strategies, the potential strategies, probably we can think about how we can induce tolerance or how we can make the transplant recipient survive for long term. The first strategy, obviously, is conventional immunosuppressions, which significantly improve the short-term outcomes, but not long-term outcomes. And the second strategy is to induce chimerism. As we already mentioned, that many patients actually cannot get tolerance at all, and some patients may die. So each strategy had advantage and disadvantage. However, a third potential strategy, graft repopulation by recipient stem cells, would have a reverse advantage over the conventional post-transplant strategies because the graft can become self and no need for lifelong immunosuppression. But the question is that possible? Stem cells from bone marrow can migrate to the whatever areas with high expression of SDF1. Recently, clinical studies in human patients with a cardiac attack show that patients who had a higher number of endothelial progenitor cells, actually they have better long-term outcome after cardiac infarction. After transplantation, if we are able to mobilize these bone marrow stem cells, and somehow these mobilized stem cells can be recruited into allograft, especially into a rejecting allograft. Imagine that stem cell may regenerate tissues and repopulate allograft. So allograft may eventually become a chimeric allograft, a mixture of donor cells and recipient cells. Here is our hypothesis. If the limited host repopulation that is currently observed could be facilitated, it's possible that conversion to a predominantly host phenotype would permit long-term growth function without immunosuppression. In this model, we found accumulation of host-derived stem cells in liver allografts at the early time after transplantation. For example, the CK, which is a bone marrow stem cell marker. OV6 is a hepatic progenitor cell called oval cell marker. And as you can see, RT1A staining with a host MHC class 1 antigen. So, as you can see, there are host-derived CK or OV6 positive cells appear in liver tissue sections at 3 days after transplantation. And the positive cells, the number of positive cells increased significantly at 10 days after transplantation. Interestingly, compared to whole liver transplantation, small size liver transplantation accelerates the ingress of host cells. For example, you can see some host-derived cells appeared in liver tissue sections as early as 3 hours after transplantation. And the number of host cells increased in a time-dependent fashion. By 10 days, you can see patches of host positive cells appear in liver tissue sections after transplantation. The host 3 population was further confirmed by using green fluorescence protein transgenic, we call GLP transgenic Lewis rats. We use GLP transgenic Lewis rats as a liver donor. So here you can see the GLP positive, small reduced size, small livers. If you transplant into the syngeneic liver here, syngeneic recipient, white blood syngeneic recipients, by 10 days, you can see the liver graft fully regenerated, the same size as the whole livers. But the GLP fluorescence expression remained the same as the donor. However, if you transplant the same, GLP positive small livers into a wild type DA, allogenic DA recipients, by 10 days, again you see the liver allograft fully regenerate to the normal liver size. But the GFP expression was dramatically decreased. That suggests part of liver allograft probably already repopulated by host-derived GFP negative cells. Interestingly, if you give the same animals allogenic recipients with cyclosporine treatments, as you can tell here, the cyclosporine immunosuppressive treatment not only inhibit liver regeneration, but also prevent the host repopulation. So finally, we use in situ hybridization method for Y chromosome in a sex mismatched transplantation models. Confirm that host-derived cells appeared in liver allografts at 10 days after transplantation, and the host Y chromosome positive hepatocytes dramatically increased at 1 month after transplantation. We found that actual hepatic stem cells can differentiate into liver tissues first. And second, liver allograft can be repopulated by recipients. And the regenerative stimuli due to a small size allograft actually can accelerate stem cell recruitment. But both regeneration and recruitment are impeded by immunosuppressive treatment. Normally, the intrahepatic stem cells or hepatocytes themselves can regenerate the liver after injury. However, these donor cells are under attack by host immune system. Therefore, probably actual hepatic host-derived stem cells can regenerate the liver after injury. However, after transplantation, usually for our patients, we give full dose immunosuppressions to prevent rejections. So in that case, the immunosuppression prevent the immune-mediated injury and protect the host cells. Therefore, no need for actual hepatic host-derived cells to regenerate the liver. So that may be reason why we only identify a small numbers of host repopulation in humans. Based on these findings, we developed 3 strategies to promote host repopulations. First, our original idea was to give a low-dose immunosuppression to convert a severe rejection into a moderate rejection. And second, we try to give a small graft, reduce the size graft and transplantation. And third, we think if we can use drugs to mobilize stem cells from the host, that may promote host repopulation. Unfortunately, in 2008, during that time, we found one drug just approved by FDA for the use in cancer patients for collecting hematopoietic stem cells before chemotherapy and radiation. After that, after chemotherapy radiations, collected stem cells will be transplanted back to the patients. We think the balance between rejection and regeneration may result in host repopulation. So interestingly, we found that in this model, tacrolimus reacted synergistically with AMD 3100 in not only mobilization of bone marrow stem cells, but recruitment of these mobilized stem cells into the liver allograft. Finally, we determine whether there's host repopulation. As you can see, we transplant a white-type liver, which is a GLP negative, into a GLP positive recipient. By 3 months, you can see dramatically increased GLP expressions in the liver allograft. And the repopulation of host cells has been further confirmed by tissue sections under fluorescence microscope. And you can see here, there are many GLP positive cells surrounded around central vein areas. So with these exciting findings, we decided to extend our studies to large animal models. And fortunately, with the funding from Zanzam Incorporations, we are able to do that studies in large pig models. The pig we used actually from provided by Dr. David Sachs from Mass General. So these pigs are SLA identified pigs, basically MHC identified pigs. So we perform SLA fully mismatched transplantations in this case. These pigs with repeat treatment at 1 month, 2 months, and 3 months can survive for long term, over 3.5 years, until our paper was accepted, so we have to sacrifice them because it's too expensive to pay the housing for pigs. $720 per month per pig at Hopkins. And so in these long-term survival pigs, we also try to determine if the animals develop antigen-specific tolerance by using mixed lymphocyte reaction with donor and also with the third-party lymphocytes. And also we perform the skin transplantation in these animals. So interestingly, the follow-up study found that indeed these animals, although skin rejected, but did not develop donor-specific antibodies. Here I show you some biopsy study biopsy samples we studied from this pig. On the left hand, you can see it's a low dose FK of long-treated pigs. That pig survived actually 57 days after transplantation. And you can see almost 1 month, the kidney already developed a chronic rejection. There's C4D depositions. But no any regulatory T53 positive regulatory T cells in kidney biopsies. However, all biopsies from these long-term survived pigs show dramatically increased FOXP3 positive cells in kidney allografts. And there's no any C4D deposition in the kidney allograft, even after skin rejection.
One great experiment I show you here. It's skin wounds created in mice, and the result as healing process proceeds is compared in this case with just a normal skin biopsy, and then the controls, and this is the combination therapy. And you can't tell too much, but it turns out that by massive trichome, there's a whole lot of fibrosis and scar tissue, and essentially these wounds heal as you would expect. A 5 centimeter wound without any coverage, just treatment for letting it heal. They form scar tissue. But with the combination therapy, here you are, no fibrosis, minimal fibrosis, hair follicles, and by 15 days, instead of irregular thin scar tissue, it's good solid skin with hair growth. And if you're not convinced by this, this is the microscopic count of the hair follicles, and there are very few in the scar tissue. But with the combination treatment, the wound heals not only 25% faster, but much more normal. So I'll state that for any inflammation or other cellular damage, including but not limited to rejection, stem cell mobilization and wound uptake produces faster, more normal healing. You have a whole lot of stem cells now attempting to do the right thing but failing, causing the fibrosis. And the reason is that there are some DNA damage from the previous alcohol feeding prior to the point of transplant. And it turns out that you can identify this 8-OHDG DNA damage adduct from oxygenation of the DNA in the mitochondria, in the stem cells of the alcohol-fed rat. And this damage is present far more in the stem cells from the alcohol-fed rat. So this is an example, and this is still very theoretical, that disease, in this case fibrosis after a liver transplant, can be caused not because of something intrinsic in the organ, but because of something caused by abnormal stem cells.
In summary, we have developed a safe new stem cell mobilizing strategy that enables long-term liver allograft survival in strongly rejecting rat string combination, and long-term kidney allograft survival without sustained immunosuppression in small animals and large animals by using a combination of 2 FDA-approved drugs. So how that works? So we think probably AMD 700 treatment increased ability, the availability of host stem cells in the circulation, while low-dose FK treatment enrich these mobilized stem cells and the regulatory T cells in the allograft. The recruited stem cells probably can promote regeneration and or repair of damaged the allograft, while infiltrating regulatory T cells probably help to create a local tolerance or immunosuppressive environment. So that results in allograft clearance and acceptance. So we think the tolerance can be induced by induction of allograft chimerism.